Good afternoon. Welcome to Qiagen's workshop here at the European Microbiome Congress. I know we're all very excited to be here, be able to share some of our tools for microbiome research with you. My name is Dominic O Neil. I'm the Director of Microbiome Product Development at Qiagen on the life science side of the business, so I'm responsible for sample extraction business. I'll be giving a talk here on a streamlined workflow to analyze changes in the gut microbiome with age. So as I said, I'm going to be presenting here on a workflow that we use, that we set up to analyze changes in the gut microbiome with age. I'll talk a little bit about the microbiome. I'm sure we're all familiar with the importance of it. And then also a bit on inhibitor removal technology, which is the Qiagen technology underlying our sample extraction kits and their particular use in microbiome samples. And then for the actual small study that we did, focusing in on the QIAamp Power Fecal Kit automated on the QIAcube, followed up with Qiagen library preparation for both 16S and whole genome sequencing, and analysis using the CLC Microbial Genomics Pro suite. So here for the introduction, we're all aware of the importance of the human microbiome and that we are vastly outnumbered by the organisms that live on us, in us, with us, and importantly that the gene content of our microbiome outnumbers our own to a vast degree. And multiple studies have shown that those microbiomes have a dramatic effect on our health, and particularly when that mutualistic relationship is disturbed, we can come and have disease. These are associations that have already been made between human microbiota and disease. In the gut, which is probably the best studied at this point, you have intestinal infections, obesity, IBD, diabetes, cancer. There's increasingly studies on gut-brain axis as well, in that the contents of our gut microbiota can affect our mental health. And then of course, airway, urogenital, blood, oral, all have strong indications that the microbiome has major effects on human health. I'll be focusing mostly around gut, obviously. And here's a brief introduction onto microbiome samples and microbiome technologies. So everything really does start with the sample. If you have a microbiome sample, if you're not able to extract the analyte of interest from that, you're not going to get good data to be able to find any new insights. And for things like gut samples, it's particularly difficult to extract DNA, RNA. I'll go into that in the next couple of slides on IRT. But there's really two major questions that we're asking in microbiome studies. Asking which organisms are present, what's their relative abundance, understanding the communities and what's present. For that, we're mostly looking at DNA, primarily around 16S sequencing, which will tell you about bacteria. If you do whole genome sequencing, you can get more information on fungus, yeast, viruses. From whole genome sequencing, you can also start to get some information on functions of the community that are present by looking at the gene content. And then of course you can dive into the functions of the community a bit more on RNA, by looking at gene expression, and also in protein small molecules when you're looking at metabolomics and basically the results of what's coming out of the microbiomes that are present. In this talk, I'm really focusing on the DNA extraction side of things. So we did both 16S and whole genome sequencing, but only on DNA. But nevertheless, Qiagen offers methods for RNA for protein extraction so that basically any microbiome question can be answered. And sample prep is really critical for microbiome studies, even more so than for some other studies, particularly because in the gut microbiome, when you're starting with a stool sample, you have a very inhibitory sample. There's a large amount of small molecule inhibitors in stool that co-purify with DNA or RNA, and which will decrease the efficiency of your PCR, your NGS, and they'll bias the results that you get downstream. And that's one of the things that we have worked very hard to eliminate, and I'll talk about on the next slide around IRT. The other really key thing to think about in terms of microbiome studies is cell lysis, Obviously, microbiome communities are complex, and gram-negative, gram-positive samples are very differently lysable, and so that's the other aspect to the microbiome kits that we offer, where we make sure that we do a very efficient homogenization of our microbiome samples in order to make sure that you're not getting any bias and only finding the stuff that's easy to lyse and easy to find. The other things up here on the list, I'm not going to go through them one by one. They are issues that come up with basically any sample preparation, and they're honestly quite well solved by Qiagen's expertise in the sample prep area. So as I said, I wanted to focus a little bit more on this co-purification issue. And so we use cell lysis, so we're doing a bead beating step to break open the cells. Out of stool, in particular, you're going to get a lot of small molecule inhibitors that are coming into your eluate. They co-purify with DNA. They're very similar to DNA, which is why they're such potent inhibitors of your downstream application. They will bind to enzymes, or they will bind to your DNA and prevent that from being amplified, or they'll scavenge important cofactors for your enzymatic reactions. And so it's quite critical to get rid of them. The major problem with them is that some of these things, some of these complex, in case of stool, it's polysaccharides, bile, bilirubin, and heme are the most important inhibitors. The problem is that even when you have what looks like a very clean extraction, you can still get a result like this. So in this case, what we're looking at is extraction from stool samples run in duplicate. So it's the same sample run 4 times, 2 of them with the IRT, the inhibitor removal technology from Qiagen, and 2 without, but all other steps were the same. And you see PCR amplification curves here. So you can see that when we do IRT, we're getting nice, clean amplification curves that come up strongly around a CT of 12 or 13. And when this IRT is removed, it's quite delayed. In one case, basically totally inhibited, where you're probably not even going to count that as a true amplification. And in the other case, also just not nearly as clean of an amplification. And these will cause bias in your downstream applications. Like I said, this is essential because on sort of normal quality specs, you won't necessarily see that these inhibitors are present because they're too similar to DNA in many cases that they'll still show up. So to sort of work out that we can do an entire microbiome study using Qiagen solutions, we did here a semi-automated DNA isolation. We chose the Qiamp Power Fecal Kit. This is a kit that incorporates the inhibitor removal technology. It is also automated on the QIAcube. For those of you who are not familiar with the QIAcube, it's a benchtop instrument. It's a very plug-and-play instrument. It automates the spin column procedure for basically all of the Qiagen kits. We have 130 standardized protocols on the instrument and can customize it very easily. And so it gives you a very hands-off automation of our spin column kits. In the case of the Qiamp Power Fecal Kit, it can take here, you start with your sample collection, you will do the microbial cell lysis off-board as usual. We take our power bead tubes, in this case a bead beating step, to break open all of the microbial cells that are present. And take the supernatant from that, you can put that on the QIAcube, and the QIAcube will go through the inhibitor removal technology as well as a spin column-based DNA isolation in a totally automated way, and you'll end up with eluate, which is appropriate for the downstream applications, whether it's whole genome sequencing, 16S sequencing, or PCR. And so the study that we looked at, we decided that we wanted to look at aging. This wasn't a full-blown study. This was something more that we thought it would be interesting to see what kind of results we came out of this. Aging, of course, we're familiar with what it is here as a definition. It's the time-dependent decline that affects most living organisms. We have these 9 hallmarks. But what's interesting about it is that there's more and more studies now showing that the gut microbiome strongly affects the phenotype of aging and the effects of aging. And then there's, of course, interest in seeing whether or not regulation of the gut microbiome would be possible to slow down some of these effects of aging. And of course, the first aspect is simply understanding what's there at different ages. So we had 10 samples. We had 10 aged samples and 10 young samples. So from the old cohort, the ages ranged from 65 to 77, a fairly even split, 3 female and 2 male. In the young samples, it was 2 female and 3 male. The age range there was from 28 to 49, so the 49 is perhaps borderline as to which group it goes into. But you can see that out of all of the samples, up on the gel above, you can see that we got nice high molecular weight DNA coming off of the QIAcube, and certainly a sufficiency of yield from all of the samples here, around 5 to 10 micrograms out of all of the samples coming through. There's perhaps an indication that you're getting a higher yield from the young samples rather than the old samples, but I'd be hesitant to read too much into that data right now. It's likely that the intrasample variability is going to be higher than here in between the groups. So the DNA is high quality. Here are the nanodrop specs. You can see our 26280s are ideal, right around 1.8, as are the 26230s. And then in addition, as I said, this isn't a 100% reliable indication of whether or not inhibition is gone. So we always do an additional test where we look for inhibition in a PCR. The QuantiFast pathogen kit from Qiagen has an internal control present. If you take your sample eluate, spike it into that internal control, in this case we're spiking 5 microliters of eluate into a 20 microliter reaction. And then you measure the delta CT of where that internal control is amplified, you can get a very nice quantitative measure of how much inhibition is present in your sample. In this case, we could see that there was absolutely no inhibition present. All 10 samples had either absolutely none or less than a 0.2 change in CT after our extraction, so we're very happy with the samples and can move them straight into our downstream applications. So as I said, what we did with this, we did 16S library preparation as well as whole genome sequencing. 16S, for people who aren't intimately familiar with 16S already, it's the amplification of the 16S ribosomal RNA gene. It's about a 1500 base pair gene that's characterized by having constant and variable regions. And the idea of 16S sequencing is that you place primers in your constant regions, which are very similar between all bacterial species, amplify through a variable region, and then in your sequencing, you can take those variable regions, map them back to the bacteria that are present, and get a good measure of the diversity that's present. For this experiment, we took the V3 and V4 regions. It's relatively commonly used. We used the established 515F and 806 reverse primers from the Earth Microbiome project for this. We took 50 nanograms of DNA into our PCR, ran 22 cycles. And after the PCR amplification, the adapters and indexes were added using the QIAseq one-step amplicon kit. This is a kit that performs a single half-hour reaction that does end repair A-tailing and adapter ligation, leading to a very, very fast library prep coming off of this. We did paired end sequencing on Illumina. One thing that I want to point out about this is that there's the 2 samples there that are blanks. Those are actually water samples that were run on the QIAcube with the same kit and then also subjected to the same 16S PCR. We did that as a control to make sure that we're not getting any cross-contamination coming off of the QIAcube, as well as no contamination coming through our samples or through our PCR reagents. You can see here that we have no amplification in the blanks, so we can be confident that we have nice, clean results and only amplified the sample that we're interested in. For whole genome sequencing, we used a different kit. We used the QIAseq FX DNA library kit. That's a kit that accepts a fairly wide range of input DNA. You can go from a nanogram to a microgram of DNA and put it in. And that has a single tube reaction that does fragmentation and repair and so on. And that has a customizable fragmentation based off of your fragmentation incubation time. Depending on your input amount and the size that you're interested in, you can use the table to select the time you want, and you'll get a fragment size as desired. We selected 350 base pairs and got a very nice result out of that. And you do adapter ligation, cleanup, and again, it's fully compatible with Illumina sequencing. So that's what we did on the these two sides. On 16S, we got 22 million paired end reads, 85% over Q30, most of them assigned to an index, and a nice even spread across our 10 libraries. And whole genome sequencing was even somewhat better. So we were quite happy with our results. It worked all pretty much on the first try without any optimization needed. And then we took the data coming off of the MiSeq and plugged it into the CLC Microbial Genomics Pro to do our data analysis. So I'm not going to talk about this too much since we have two other talks which will be able to talk about the Pro Suite in far more detail than I'm able to. But for us, what was important as sort of end users of the program was that it was a very easy plug-in into the genomics workbench. It had basically all of the modules, the plugins and analysis tools that we needed to do basically full analysis of our samples. It was a very easy to use graphical user interface, and the non-bioinformaticians in my group were just as happy to use it as the bioinformaticians were. So for that, it was very, very helpful. And so our workflow in the Microbial Genomics Pro Suite looked something like this. We took our raw reads from the MiSeq, imported them in, did trimming and quality filtering of all of the reads. We merged our paired end reads, added in databases that we were interested in. For 16S, we were looking at green genes. For whole genome sequencing, we were looking at gene ontology and PFAM. And we did OTU clustering. You can do open and closed OTU picking, table filtering, tree building. And then you can look at alpha and beta diversity. You could also look at phylogenetic diversity, the unifrac distances, and do statistical analyses on any of these measures that you're looking at. On the metagenome assembly, we can do both direct mapping as well as de novo assembly, identify genes and coding regions, and build functional profiles off of that, and look at the gene ontology and see which functions are present. So I'll jump right into some of the data. So here's an example of what came out of our 16S OTU and taxonomic assignment. And this was interesting. There's some differences that you can see just based off of looking at the thing. We've got old over here on the left and young over here on the right. You can see that there's a bit more here of the ruminococcus in all of the young samples. Here, the Bacteroides is relatively similar for all of them. Overall, based off of the number of small amounts up here, as perhaps an indication that there's a little bit more diversity in the young samples rather than the old samples. That you can see that more clearly on a later slide. I think it's a little bit difficult to read out of here. Another interesting thing that you could see here is these dark blue bars that show up are actually Akkermansia, which is thought to be associated with a healthy gut microbiome and sometimes used also in probiotics. So that'd be interesting to trace back possibly to diet here and see whether or not this giant block of Akkermansia is here, is somebody attempting to modify their gut microbiome. And so we got a lot of interesting data out of this. Looking at alpha and beta diversity from the 16S, we saw also data actually pretty much in line with what we were expecting. Here, so for people who aren't familiar with alpha diversity, you're taking the number of OTUs that you detect here on the y-axis, plot it against the read depth. As read depth increases, you find more and more of these OTUs. And the higher this curve goes, the greater the diversity there is in the sample. This eventually plateaus out once you get into incredibly high read depths. But for the most part, you're looking at sort of this inflection point to see where the most useful point of sequencing is. What we found is that the young samples, these 5 pinkish samples, are the young ones, and these dark blue ones are the old samples. And it's clear that in all cases, the young samples had a higher alpha diversity than the old samples. This is something that has been seen before. And then there's especially since a higher diversity is associated with better health outcomes, this would likely be something that would be very interesting in aging studies to see whether or not you can keep diversity higher throughout aging and thereby perhaps influence better health outcomes. If you're looking at beta diversity, this is distinctions between samples rather than within the sample. And here we have a fairly good clustering across the samples. We have the young samples clustering primarily along this principal component axis, the old samples clustering down over here. We have one sample which clusters, a young individual that clusters with the old one. It's notable that that is the old young sample at 49. So it could also be that the arbitrary old young split that we chose is obviously at the wrong spot, or potentially that this individual has a microbiome that's undergoing a shift faster. Obviously there's not enough data here to say, but it's clear that this one is a bit of an outlier from all of these others. This is a significant difference by Permanova. This was all unweighted unifrac right now, but you can also do weighted unifrac, generalized, Bray-Curtis, Euclidean. You have multiple options to look at the data how you would like to. In all cases, this is highly customizable. You can change colors, scales, whatever information you'd like to display in these things. On the whole genome sequencing side of things, we did, as I mentioned, functional genomics, looking at gene ontology, and here we're just looking at some of the pathways that are upregulated in samples from young individuals versus old individuals. These are the top 25 pathways that we found to be differentially present. There's some interesting patterns that show up in young individuals. There are quite a few DNA recombination, nucleic acid DNA binding, DNA metabolic activities, DNA mediated transposition, sort of nucleic acid activity here as well, nucleic acid metabolic processes, which seem to be more active in the younger individuals, as well as a mixture of metabolic processes, which are all upregulated in young individuals. I'm not enough of an expert here to say what the significance of it is, but the pattern is quite striking to see. So you can pull out some very interesting information in this sense as well. You can do tree building as well from the functional assignment. We did that here as well. We see very similar results to what we got in our beta diversity, in that the old samples primarily cluster together. I believe this is our old young sample, which clusters with these other ones. And then here the young group clusters here as well. So I'm going to wrap up at this point. Just to summarize, we did a semi-automated isolation of DNA using the Power Fecal Kit on the QIAcube. Pulled that DNA directly into our downstream, where we did both 16S and whole genome sequencing using QIAseq kits. And that was then all analyzed using the Qiagen Microbial Genomics Pro Suite for both the 16S and whole genome sequencing. And we got some very interesting results out of this. And at this point, I'd be happy to take any questions.
Qiagen.
Sample to insight.